Moyen, this room is almost the size of my country. It's impressive. Yeah, for those that don't know, Luxembourg is a small country, 600,000 people between Belgium, Germany, and France. And we do research. I will only show a bit of research from my group. The rest of the lecture is more an overview on the landscape of models that we do have for the pulmonary system. I will show you really a lot of slides from different companies. And because of this, I choose today to have a conflict of interest statement expressing explicitly that I do not have any interest in any of these companies. But you may think so. What I show is not an endorsement for any specific technology, for any specific company. And I also want to say that I have got from some of the companies, so to say, official slides, and some slides were simply taken from their websites. You will also see that some of the companies I mentioned are here exhibitors, and I think even 2 companies are giving talks in this session. So how far has the community come when it comes to pulmonary system? What you see on this slide is a representation of an alveolar barrier. You see endothelial cells below with some dendritic cells in the blood. You see macrophages on top of the alveolar cells. It's a nice graphical representation, but how are we really scientifically? When it comes to the lung, there's actually an interesting documentation from the JRC. The ECFAM has done an extensive literature research on cell culture models representing the respiratory tract. And what they found, and this is really amazing, is 284 different models. Now, this sounds like a lot, but if you use a different cell line in a slightly different setting, it counted at 2. If you start to combine 2 cells in a different setting, it also counted as a new model. So the number of actual models is partly slower. And most of these models have been used in an academic setting and very often in do it once, publish once, and never do it again setting. Well, I will try to show you things that are really at the market or very close to the market and can be used by industry. There's another kind of disclaimer or baseline that I want to set here. We often do cell culture in a classical way, submerged. And I really want to make here the statement, when it comes to the lung, we have to work at the air-liquid interface. It's not possible if you think about particles to work submerged, because you know protein corona, etc. You test something which is not human relevant. And if you already have to work for nanoparticles at the air-liquid interface, why not work for chemicals too? Because this allows you to better transfer and compare data between particles and chemicals. It's a long list. The only reason I want to show is that air-liquid interface is not something exotic. Most of them are from the last years. Some of them are already 20 years old. And they have been used for airway cells, for alveolar cells. You can use them in 3D culture settings. There are human primary models commercially available. And even you can have air-liquid interface, and I will show you some examples, in the form of organ on a chip. So there is no reason to work with lung in a submerged setting. Before I go to all these commercial things, I will show a few slides from the work that we do in Luxembourg. On the right side, you see a schematic representation of how an alveolar epithelial area looks like. You have epithelial cells type 1, type 2. You have capillary cells, and of course, you will find some immune cells. And that's what you try to do. We established transwell insert models, which you see on the left side. Again, in a transwell insert with a membrane of different pore size, depending on what you want to do. Endothelial cells, epithelial cells, and immune cells. And this allows you, and this is indicated with this orange dot, to study, for example, particles in a natural way without any interference with serum components. When it comes to these cell models, and especially when you, for example, switch for the first time from submerged culture to air-liquid interface, you have to do some characterization steps. One of them, you want to be sure that your alveolar cells produce the surfactant, that your bronchial cells produce mucus. There is a very easy test. If you culture them and they would not express surfactant, the cells would anyhow die. But I also want to show that the cells really produce surfactants. And what you see here is a simple test. And on the upper part, you see alveolar cells A549. You use a specific phthalate solution with a blue dye. And if it's a small spot, you know you have surfactant expressed at the surface, not intracellular, but really at the surface. And as a comparison, you see the endothelial cells that we use in our model. You see a big spot showing that, of course, there is no surfactant produced in an endothelial cell line. Just one easy step to show that your model is properly working. We have macrophages in our system, and I want to show you also that the macrophages do what they're supposed to do, namely to take up, to phagocytose particles. In this case, we used 50 nanometer large silica nanoparticles. They were rhodamine labeled. We sprayed them for 30 minutes. Cells were incubated, the models for 48 hours. And then we stained the cells for for confocal microscopy. What you see is in blue the nuclei, in red the cells, and in green antibody against CD54, a marker for mature macrophages. And you see several of these macrophages. In the inserts BC, in the middle of the picture, you see that these macrophages, the green marked cells, have white spots in the cytoplasma. And these white spots are accumulations of the silica particles that we have sprayed 2 days before. So the macrophages have exactly done what they were supposed to do. They've taken up the silica nanoparticles, and you also see no white material outside of the macrophages. So they have really cleaned the surface of this transwell insert. Now we have a complex model, and I want to show you also kind of the beauty of complexity and what you can do with a complex model. And in this case, we sprayed diesel exhaust particles, diesel on the cell culture, on the epithelial site, like you would inhale. And what I will show you as a result is effects that we have seen on the endothelial side. So whatever you see is due to either material coming from the diesel particles that have migrated through the pores into the endothelial cells, or second messengers, chemokines that have come from the epithelial side that induce something on the endothelial side. The material was sprayed using exposure equipment from a German company, Vitro, which is also exhibiting here. And that's what we saw. So we sprayed our cell culture models at what we think were realistic concentrations. And the data I show you again are from the endothelial side. So I do not talk now about the epithelial side, let's say the direct exposure. Everything I show you is indirect exposure. And if you see in the red box, there were no statistically significant differences in cell viability between the exposures and between the air-liquid interface. So the culture is fully viable. That's nice to see. Then you should never do that. I show you so many digits in the next box, and you don't have to read them because there were no differences in any of the genes measured on the endothelial side. So that's easy. Forget about the numbers. No effects due to the exposure. And the next I don't show, but I zoom immediately in because that was the last thing. And here it becomes really interesting. 4 hours after we had exposed the tetraculture to diesel exhaust particles, we see something very interesting. And this is the lower left quadrant, the green picture that is of interest now. That's immune staining against NRF2, which is a cytoplasmatic receptor that circulates between the cytoplasma and the nucleus. And upon exposure to aliphatic chemicals like benzpyrene, which is present in diesel, it binds to the DNA. And what you see is on the left, there's a little bit of green in the nucleus because it's, as I said, a shuttling receptor, but on the right side, after exposure, the green dye accumulates in the nucleus. That's an indirect effect, a very early effect, and this will turn on genes later on, and as a result of indirect exposure. Now coming to complexity, I have 2 more slides. Why is complexity so interesting? We have 4 cell types in our model. Is this really necessary? What is the added value? We did this with an industrial partner, Philip Morris International in Switzerland. And there was no exposure at all, what I show you now. This is just epithelial cells alone. And then we looked into the active gene networks. Then you add endothelial cells, and again, you look into the active gene networks in the 2 sides of the transwell. You add the immune cells, and again, you look into the active gene networks. What do we see? First, on the left side in blue, you see the results from a 3D model, and in violet, pink, you see the results of a 2D, and the first observation is that the 3D model has higher bars than the 2D model. Now, the y-axis of these 2 figures is the similarity between an in vitro model in respect to active gene networks and the human non-exposed in vivo situation. This is compared to patient material from surgical operations. So you see that the 3D is very similar to the human in vivo situation, and within the red circle you see also something interesting. That's this tree culture, meaning epithelial cells, endothelial cells, and macrophages, or in addition the mast cells. And what you see is that the addition of the mast cells does not add anything relevant anymore, because you are on a kind of asymptotic curve. You are at around 80% similarity to the human in vivo situation. So that's why we kicked out the mast cells from our system, because it has no added value. You only have consumable costs, time costs, etc. So now I come to the second part, kind of my presentation. Some of the commercial cell systems, simply to show what is available for the community. One company, Swiss French, Epithelix, they have primary human material, and they can represent the nose, the trachea, the bronchi. You get them in different well sizes. Maybe I go direct to the next one. If you know human bronchial histology, that looks very much like a human bronchial tissue would look like. You even see the cilia. The cilia are also beating in the system, and the beat rate can even be used as a readout. What is also nice is you have certain diseases available, meaning material coming from diseased patients, such as asthma, COPD, cystic fibrosis, etc. Second company, US based for Europe in Slovakia. They have similar models. They also have an alveolar model, and they have also tissues for organs which are relevant for the lung but are not lung. If you think about inhalation, of course, having a model also for the eye, the cornea is of interest, because you will inhale what you also get in your eye. So you can have a quite complex and complete exposure model. One model which is very endpoint specific that I want to show, this is now when it comes to respiratory sensitization. Swedish company SensaGen, they have the GARD Air. It's a model based on one cell line and an AI readout of a set of genes, which allows to differentiate between chemical sensitizers and chemical irritants. Another company, they are also here in Screenex, and they are specialized in producing certain cell types. Why is this of interest? I showed you data and cells from A549, and A549 are often criticized of having not fully functional tight junctions. This is a cell line that has fully functional tight junctions. The cells develop, or the models develop, a very high transepithelial electrical resistance, which the A549 don't do. So dependent on what you want to study, this may be a very important feature that you want to have. Another aspect that you can have, Immuno1, it's a British company. They provide immortalized alveolar cell line in combination with immortalized alveolar macrophage cell like cells. These models are available both at the air-liquid interface, but also liquid-liquid, meaning submerged, and in a 24-well format. They are now since 2 years commercially available. A bit more recent, Japanese company, Hailang, they have developed for the first time, to my knowledge, IPS-based model that is also available and may have promising features as it is different from the cell line and tumor cell line based models that I've showed so far. Some of these models are also available as a lung on a chip. That's one of them, Emulate. They are also here. You can culture them in a similar system that we have seen at the end of the liver talk, but you can also culture in this system lung cells. You can have stretch as an additional factor. Another model from Mimetas that's quite complex. You see, it's almost like a multi-well organ on a chip system. You can load it with primary human cells, dependent what you want, alveolar, endothelial, and you can also add stretch again. That's lung on a chip model. The company is also here. You can combine it with inhales incubation, and then next speaker will show you about this. What is missing is this, the pressure. Company is here. It's a pressurized system. It's a kind of 96 well plate format, and you can add pressure and the system is breathing. With this, I come to my last slide. It's very important that you, if you develop something new, that you properly characterize your system. Think about complexity, how complex you want to be. If you go for the lung, you should work at the air-liquid interface. Keep in mind dosimetry. The smaller your models start to become, can be very difficult. And all models have advantages and disadvantages. So it's your research question that will drive the choice. For those that have still time, in 3 weeks, there's a very interesting conference in Europe, in Nice, Live 2020, where the lung model environment will be collected. And with this, I finish. Some more Luxembourgish words. Merci vielmals. And I'm happy to answer questions.